Hello viewers, I am Dr. Raviul. I work as a lecturer of pathology in a medical college hospital and I am making this video for my students and also for you. Hope someone finds this helpful. Today's topic is on immunologic tolerance. In this lecture, we will learn about the definition of immunologic tolerance, the factors determining immunologic tolerance, and we will also talk about the types of tolerance that will include discussion on central tolerance and peripheral tolerance. Okay, a lot of topics, so let's begin. First question, what do we mean by immunologic tolerance? As written in your textbook, it is the phenomenon of lack of responsiveness to a specific antigen that could otherwise evoke an immune response. Okay? So let's repeat this definition one more time and then I will explain this definition. Immunologic tolerance is the phenomenon of lack of responsiveness to a specific antigen that could otherwise evoke an immune response. Now let's explain this definition, and we will explain this with the best example of immunologic tolerance, and that is self-tolerance. Always remember, self-tolerance is the lack of responsiveness to an individual's own antigens, and these same antigens can be considered foreign if they were transplanted to a different host. Now to explain immunologic tolerance, you can see that I have drawn a simple cartoon and it is showing two interesting scenes. On the top we can see a dendritic cell. This dendritic cell is very happy because it is presenting self-antigen to a nearby T lymphocyte. And the T lymphocyte, when it is presented with such self-antigen, is also looking happy because that T cell knows that this is part of our own body, so no response is required. And this is an example of self-tolerance. Now let's focus on the bottom image. What can we see here? Here, the dendritic cell is not very happy because it is presenting something foreign to the T lymphocyte with the help of MHC molecules. And when the T lymphocyte is presented with such foreign antigen, it is reacting. It is also not happy. It must initiate immune response. So this is an example of immune response, and the previous image, or the image that is drawn on the top of this slide, was an example of immunologic tolerance. So always remember, the best example of immunologic tolerance is that of self-tolerance, and it means our body is tolerant to our own antigen or to self-antigen. So now that we have defined immunologic tolerance, now let's move on and talk about the factors that determine immunologic tolerance in our body. They will include immunologic maturity, antigen structure, antigens cross-reacting potential, pro-inflammatory signals, duration of antigen exposure, etc. Regarding immunologic maturity, always remember we are tolerant to those antigens that were present during the process of immune maturation that occurred during early development. Regarding antigen structure, always remember that simple molecules, for example, small proteins, are more likely to evoke or initiate tolerance compared to complex molecules like polysaccharides. Regarding antigens cross-reacting potential, always remember that although T cell receptors and B cell receptors are highly specific, sometimes they do confuse one antigen with another antigen. And what will happen? As a result, there will be response against foreign antigen that is inappropriately targeting self-antigens, and that will also result in a condition that is known as autoimmunity. Now, we will talk more about autoimmunity in another lecture. For this lecture, just remember that occasionally T cell receptors and B cell receptors can confuse one antigen with another, and as a result, the response that was targeted against foreign proteins may inappropriately target self-antigen, and that will result in loss of self-tolerance or loss of immunologic tolerance. So that is an important factor to remember. Pro-inflammatory signals are also important factors in determining immunologic tolerance. And regarding duration of antigen exposure, always remember tolerance is best maintained when antigen, which is inducing the tolerance, continues to be present. So now that we have talked about the factors that determine tolerance in our body, now let's move on and talk about the types of immunologic tolerance. So this slide is showing the types of tolerance. They are of two types, central and peripheral. First, let's talk about central tolerance. What do we mean by central tolerance? It is self-tolerance that develops in central lymphoid organs. Now, whenever you mention this term, central lymphoid organs, the examiners are very fond of asking you what are the central lymphoid organs or primary lymphoid organs. And always remember, thymus and bone marrow are our central or primary lymphoid organs. So, self-tolerance that develops in central lymphoid organs are known as central tolerance. Here, immature self-reactive T and B cell clones that recognize self-antigens during their maturation in central lymphoid organs are killed or made harmless. So now let's talk about central tolerance in T cells. Now, antigen receptors of T cells are known as TCR or T cell receptors, and these are generated by somatic recombination of genes in a random manner inside the developing T cells. Since somatic recombination is occurring randomly, T cell receptors capable of recognizing self-antigens are also generated constantly. However, we don't want T cells that can react with self-antigens. So, when immature T cells with such T cell receptors encounter self-antigens in the thymus, many of those T cells will die by apoptosis, and this is known as negative selection. So whenever you mention this term, negative selection, the examiners are very fond of asking you about the selection processes of T cells and B cells. Now always remember, lymphocyte development occurs in multiple steps or checkpoints. At each checkpoint, the developing cells are tested and the cells continue to mature only if a preceding step has been completed successfully. So these checkpoints are successful production of one polypeptide chain of the two-chain antigen receptor. Recall that antigen receptors of B cells and T cells, they are composed of two chains, and the first checkpoint is successful production of one polypeptide chain of this two-chain antigen receptor. The next checkpoint is assembly of a complete antigen receptor. So now we have both chains together. Then the other checkpoints are positive selection and negative selection. So first, let's talk about successful production of one polypeptide chain of the two-chain antigen receptor. Now, pre-T cell receptors and pre-B cell receptors contain only one of the two polypeptide chain that are present in a mature antigen receptor. Developing T cells that successfully rearrange T cell receptor beta chain gene will synthesize T cell receptor beta chain protein and assemble a pre-antigen receptor that is called pre-TCR. Similarly, developing B cells that successfully rearrange their immunoglobulin heavy chain genes will express the mu heavy chain protein and assemble a pre-antigen receptor that is called pre-BCR. The assembled pre-antigen receptors will provide signals for survival, proliferation, the phenomenon of allelic exclusion that we will talk about shortly afterwards, and further development of T and B cells. The next checkpoint is assembly of complete antigen receptor. Lymphocytes that have successfully passed the pre-antigen receptor checkpoint will go on to rearrange and express genes encoding the second chain of the BCR or TCR. They will express the complete antigen receptor while they are still immature. Cells that express useful antigen receptors will be preserved. Cells that strongly recognize self-antigens will be eliminated or induced to alter their antigen receptor, particularly in case of B cells. So now let's talk about positive selection. Within the thymus, cells of the thymic stroma express both class 1 and class 2 MHC proteins, and with the help of these class 1 and class 2 MHC proteins, they can present self-antigens to double positive thymocytes. If double positive thymocytes bind to major histocompatibility complex molecules that are loaded with self-peptides with low affinity, they are given the necessary survival signals through their T-cell receptors, and they will divide into a population of clones. This is known as positive selection, since only the T cells that bind to MHC molecules loaded with self-peptides with low affinity are chosen to survive. Double positive thymocytes that fail to recognize self-MHC at all will not be encouraged to mature. This is known as failure of positive selection. What do we mean by negative selection? As double positive thymocytes that have survived positive selection begin to move towards the thymic medulla from the thymic cortex, the second selection process will occur. Thymocytes that bind too strongly to self-antigens are deleted by a process of programmed cell death called apoptosis. Now, I have an entire lecture on apoptosis, so you can also look into that lecture after finishing this lecture to know more about apoptosis. Now, one thing we have to remember, although double positive thymocytes co-express CD4 and CD8, the cells are directed to express only CD8 if their T-cell receptor binds to class 1 MHC molecule, and only CD4 if their T-cell receptors bind to class 2 molecules. At this point in development, the thymocytes are referred to as being single positive. Now, always remember regarding the selection process of T cells, this selection process is very rigorous. A total of 95 to 99% of all T cell precursors entering the thymus are destined to die. Only those with T cell receptors appropriate to protect the host from foreign invaders will be permitted to exit to the periphery. CD4 positive cells that recognize class 2 MHC are destined to become helper T cells, and CD8 positive cells that can recognize class 1 MHC are destined to become cytotoxic T lymphocytes. Now, whenever we are talking about negative selection in the thymus, the examiners are very fond of asking you, how do these thymic cells display self-antigens? Or how do these thymic cells present peptides that are found outside the thymus, for example, that are found in the pancreas or lungs or salivary glands, etc. This is a very interesting question, and the answer is a transcription factor known as autoimmune regulator causes enhanced and random synthesis of these self-proteins in the thymus. This causes the thymic cells to display peptides from proteins that are usually found outside the thymus. Now, what will happen when there is mutation in this transcription factor? Mutations in the gene encoding AIRE protein will result in a condition known as autoimmune polyendocrinopathy. And what will happen here? Since there is mutation in the gene that used to encode AIRE protein, so the thymic cells will fail to display proteins or self-proteins, and as a result our immune system will not be tolerant and our immune system will inappropriately attack multiple organs. So now let's talk about central tolerance in B cells. B cell clones also go through negative selection. This occurs primarily in the bone marrow. Always remember that tolerance in B cells is less complete when compared to tolerance in T cells. B cell precursors that strongly bind to self-antigens are subjected to clonal deletion by apoptosis, or they are subjected to energy. However, such B cell precursors can get 1 second chance to escape this fate by a process that is called receptor editing. This is very important. Always remember that receptor editing only occurs in case of B cells. Receptor editing does not occur in T cells. So, what do we mean by receptor editing? And you may ask me, Dr. Rabiul, which receptors are we talking about? We are talking about B cell receptors, and recall that we know that B cell receptors are in fact immunoglobulin. Recall from my 2 lectures on antibodies in the immunology lecture series, we know that immunoglobulins are made up of 4 chains. they are two identical heavy chains and two identical light chains. We had also learned that these chains contain variable region and they also contain constant region. So the receptor editing is occurring in the variable region of light chain. Recall that we have light chain kappa and lambda, and the gene that encodes the kappa light chain, those gene clusters are located in chromosome 2, and the gene clusters that can encode the lambda light chain, they are located in chromosome 22. And again, we have kappa and lambda gene from our both parents. So we have a lot of variants or alleles that can make the light chain. So what is happening in receptor editing? In this process, an alternate VL combination is made by using an unused light chain allele, and that can replace the previous light chain allele, and that will create a new light chain. And since the immunoglobulins are composed of light chain and heavy chain, if we alter the light chain, we will in fact alter the entire immunoglobulin. And since the immunoglobulin is acting as receptor here, so due to that alteration in light chain, ultimately we will create a new B cell receptor or a new immunoglobulin M receptor. And this is receptor editing. Whenever a B cell precursor is reacting strongly to self-antigen or self-protein, that B cell precursor can be given a second chance to escape apoptosis and energy, and that is achieved by receptor editing, where it is changing the light chain of its antigen receptor or B cell receptor, and that is creating a new B cell receptor that will not act so strongly with self-antigen. And always remember that 25 to 50% of circulating B cells have undergone receptor editing. So now that we have talked about central tolerance, now let's move on and talk about peripheral tolerance. What do we mean by peripheral tolerance? It is tolerance that is acquired outside the central lymphoid organs or outside the primary lymphoid organs. Why do we need peripheral tolerance? It is necessary because some self-antigens are not displayed by the thymic cells, and therefore some self-reacting T cells are not eliminated in the thymus. It is also necessary for development of tolerance to harmless commensal organisms on our skin and gastrointestinal tract. So, what is the mechanism of peripheral tolerance? In this slide, we are showing the mechanisms of peripheral tolerance. They will include absence of co-stimulation, suppression of naive T cell activation by regulatory T cells, and sometimes naive T cells themselves can differentiate into regulatory T cells or Tregs during the time of their activation. And always remember, regarding absence of co-stimulation, when T cells are activated without co-stimulation, they become anergic. What do we mean by anergic? It is a state of non-responsiveness. So what is the role of T regulatory cell, or what is the mechanism of suppression by T regulatory cells or Tregs? Always remember, Tregs have high baseline level of high affinity interleukin 2 receptor. These receptors act as a sink and they reduce the amount of interleukin 2 that is available to other T cells. And by that it induces peripheral tolerance. Tregs also have high levels of cytotoxic T lymphocyte antigen 4 protein. And always remember, CTLA4 and CD28, they compete for binding with B7, but CTLA4, that is located on the surface of T lymphocytes, has higher affinity for B7, that is located on antigen presenting cell, compared to CD28. So whenever CTLA4 on the surface of T cell has bound with B7, that is located on the surface of antigen presenting cell, CD28 on the T cell surface is unable to bind with B7, and that will block the B7 CD28 co-stimulatory signal pathway, and that will result in tolerance. And also remember, Tregs can secrete interleukin 10 and transforming growth factor beta, and all these things can also result in tolerance. So this concludes our lecture on immunologic tolerance. I hope this lecture was helpful. If you like my lectures, do comment, share, subscribe, and let me know. And for my students, I will also recommend you to go through your textbooks to know more. Okay, that's all for today. Until next time, take care and stay safe. Thank you.